so one idea that's coming around is that instead of killing bacteria, which is what traditional antibiotics do, they pop their membranes, they make it so bacteria can't replicate their DNA. And so they kill bacteria, and resistant mutants then have a selective growth advantage because they can grow in the presence of the drug. So the question is whether or not, instead of continuing to do that, could we instead do some kind of like behavior modification? So for example, if we could make bacteria so that they can't talk or they can't hear, could we then confuse them so that they don't understand they're in a group and they don't turn on these virulence factors? And so there's sort of two strategies that are being worked on. The first one is to target, and the logic is the same. So the first one is to target the intraspecies system. So if you want a bacterial-specific drug, so for example, you want to treat CF, you want a drug that works against Pseudomonas, right? Because that's the cause of that disease. Then what one could do is to try to shut down very specifically this intraspecies communication system. And then hopefully what you could get would be bacterial-specific therapeutics. The alternative, which is obvious, is to go after this interspecies system. So if you wanted a broad spectrum drug that would hopefully work against lots of bacteria. So you go to the hospital, you're sick, the physician doesn't know what you have. They try to treat you for everything, right, initially, while they figure out what you have. So maybe you could instead shut down interspecies communication, and that could be a broad spectrum antibacterial therapeutic, right? And so the way that you do that is that you make bacteria that either can't make or can't detect the molecules, either through this system or that system, right? So that's where this is all going. And so we're interested to actually do that. And so then the question is, how do you actually go from this idea of, oh, if I could have a molecule, then that'd be cool, right? So how do you actually do it? And so I'm going to tell you how we've done it for this system. We've done it for the species-specific, and other people too, species-specific system too, and the idea is exactly the same, but I just picked one of these systems to show you. Okay, so you guys know that bacteria make this, what we call autoinducer 2, this second molecule that I use as these pink ovals that gets detected by this receptor and tells the bacteria to turn on genes. And so what we would like to do is to be able to inhibit that system. And so how do you go about doing that? Well, so you guys know that in my cartoons I use this pink oval to mean autoinducer 2, but of course you've seen that this is a molecule. This is a molecule with a three-dimensional structure that locks into that receptor like a locking key. And so what one does is one simply does chemistry. So we start with a molecule that looks like this, and we change its shape. So for example, you add a nitrogen, you add a sulfur, you put a ring on it, you make it a couple carbons longer, right? You simply start with the original molecule and change its shape a little bit. And what we're hoping to get is a molecule that looks kind of like the real one, that fits into that receptor and doesn't turn the key, but blocks the real molecule from being detected, right? So does everybody get that? So you can start with the molecule if you know what it looks like and just change its shape. And so we made a library of molecules like that. The other thing we did, which turned out to be actually easier, is that chemists make molecules for a living. That's what chemists do. And so all these chemists over all these years have made all these molecules, and they've put them into libraries that, like the NIH has, and you can access libraries of molecules that don't look anything like the real one. These are just molecules that chemists have made. There's hundreds of thousands of these in the library, and you could just go and ask, does any one of those, for just fortuitous reasons, happen to look good, that it locks into this receptor and shuts down quorum sensing, right? So you can either make them by rational design, starting with the one you know, or simply do a random screen for molecules that, for whatever reason, interfere with the process, right? So does everybody get that? So using our library that we made and using this NIH library, that's what we wanted to do. And so the question is, you can make all these molecules. We have access to these hundreds of thousands of molecules at the NIH. The question is, how do you get the few that actually are going to work as a drug? How do you screen through all of those molecules? And so what we thought we could do is to go back to these harmless but beautiful Vibrio harvii that make bioluminescence. So you'll remember that in the course of the studies that I've told you, we've made this Vibrio harvii mutant that only has this system 2 quorum sensing. And so remember, if we add this molecule, light turns on, which is something that we can see and we can measure, right? So remember that. So what we can do is we can just take that bacterium and one by one add all of these molecules from these libraries, one by one, and ask, does any of those molecules make it so this bacterium doesn't make light? Everybody get that strategy, right? So you can go in a week, from 300,000 molecules, right, that look, most of which are duds, of course they are, to about 1,000 that actually shut down light. And so it's really simple. Robots do this, and all they're doing is measuring light or not, right? So we took this Vibrio harvii and we did that. We went through these 300,000 molecules and we found about 1,000 that shut down light. If I dump Clorox on these bacteria, they also won't make light, right? And so we are not interested. So there's all kinds of reasons, they're poisonous, right? They pop their membranes. We're not interested in molecules that don't work through quorum sensing, right? So after we got down to these 1,000 molecules, what we wanted to say is which are the ones that are really working like this instead of just doing something awful to the cells. And so then you'll remember that we had this other, you won't remember this, you'll remember that we had this other Vibrio harvii strain, right, that only turns on light through system 1, remember? So what we do is we went from 300,000 to 1,000 molecules, and then we take those 1,000 winners from that last screen, and we put them on this strain, and now we want bacteria that make bright light, right? So we want molecules that, so now we know they're not just affecting luciferase, the enzyme that makes light. They're not just killing the cells, right? So we just go back and forth and ask for molecules that inhibit light in this context and that do nothing to the production of light in this context. And then we know we have molecules that specifically act through this second system, right? Right, so that's the logic. So that takes a week, right? I mean, don't get me wrong, it took me 15 years to make the strains, but so it took us about a week to get from these 300,000 molecules to about 20. And then when you have a number like that, you can actually do a real experiment. Because what it turns out is that no one actually cares whether or not I can turn on and off light in Vibrio harvii. That is like something that in my middle age has occurred to me. What they really, that's called a midlife crisis that I'm having. What they really care about is this, right? It doesn't matter that we can shut down light in Vibrio harvii. It matters that we can shut down biofilms and virulence factor expression in a pathogen, right? So we go from these 300,000 molecules that you could never put in animals. You could never test that many molecules, but we could test 20. And so what we did was we took a pathogen that we know requires system 2 quorum sensing to kill this animal model, a mouse in this case, and we've done this in a few other animals, right? And so what happens is if you infect the animal with that bacterium, the animal dies. And so the question is, if we infect the animal in conjunction with those winners from that screen, can we get an animal that lives? And the answer is yes. 2 of those molecules work, right? And so we went from 300,000 down to a couple molecules, and these are not drugs. These are lead molecules that are now going to go hopefully forward to be developed into drugs, right? So the lead molecules don't have nice properties that would make them last a long time in your blood system, that you would take a tiny little pill instead of a pill like this, that they can't be toxic to the humans. And so the place that we're at right now is we have these lead molecules, and then with medicinal chemists, you iterate with these molecules to make them have better and better properties that chemists know are good for making drugs. I mean, there's a whole field of medicinal chemistry that those kinds of properties are known. And so we need them to be more potent and less toxic to the humans, right, and more potent to the bacteria. So that's the place that we're at right now. But it looks pretty good as a new idea of a way to be able to treat bacteria, because it does cure the animal of the pathogen. So what I hope you think now is that bacteria talk to each other, and they talk with chemical words. They don't talk with the kinds of words we use, but they use chemicals as their words. What I hope I've brainwashed you into thinking is that bacteria are multicellular, right? So because they have quorum sensing and this ability to synchronously turn on and off behaviors, that is multicellularity. I mean, bacteria have been on the earth for 4 billion years, right? They invented multicellularity, and we think that in fact the way multicellularity works, the way these receptors look, the way the information gets transduced, it smacks of what people know about in higher organisms. So we think that it's useful to study bacteria in order to understand things about higher organisms like human beings.